Thank you for viewing this abstract entitled Rethinking UTI, Distinct Symptom Profiles Associate with Specific Urinary Microbiota. These are our disclosures. Over half of all women will develop UTI in their lifetime. UTI is overwhelmingly considered to be E. coli-centric, and little research has been done to assess how the baseline urinary microbiome impacts UTI development. The objective of this study was to assess associations between urinary microbiota and lower urinary tract symptom profiles, or symptotypes, in women with UTI symptoms. This is a sub-analysis of an IRB-approved study that included adult women presenting for UTI symptom evaluation, who responded yes to the question, "Do you feel you have a UTI?" and agreed to transurethral catheterization. Participants answered the Validated Urinary Tract Infection Symptom Assessment Questionnaire, or UTISA, and contributed catheterized urine specimens, which we assessed by EQUIC. Principal Component Analysis, or PCOA, of UTISA scores clustered participants into symptotypes based on severity and degree of bother of UTI symptoms, and biplot analysis identified symptoms responsible for variation. EQUIC results were then compared across symptotypes. PCOA of UTISA scores resulted in 6 distinct symptotypes, as shown here, and biplot analysis revealed the main UTISA components responsible for variation between symptotypes. These were the severity and degree of bother of urgency and incomplete emptying, severity of abdominal pain and lower back pain, severity and degree of bother of frequency, overall symptom severity, and the severity or degree of bother of dysuria. Some symptotypes, such as A, C, and E, complained of severe, mild, or moderate symptom severity and degree of bother for each UTISA category. The other symptotypes, B, D, and F, had increased severity and degree of bother for frequency and urgency, incomplete emptying, and frequency, respectively, compared to remaining categories. Culture analysis, represented here by a relative abundance graph, revealed that the rate of culture-negative samples was similar across all symptotypes, with a p-value of 0.13. E. coli, shown here in yellow, was also detected at similar frequencies in all symptotypes, with a p-value of 0.31. Although no single microbe was always present within samples in a single symptotype, some symptotypes exhibited an increased frequency or abundance of certain microbiota. The removal of E. coli from all symptotypes allowed us to visualize the difference in frequency and abundance of remaining microbiota, demonstrated by this relative abundance graph. These community structure differences were further explored with PCA biplot analysis, which shows that the 6 symptotypes, represented here by different colors, occupied slightly different areas of the 3-dimensional PCOA plot. This separation was based on 6 main points of variance, which were the presence and abundance of Klebsiella pneumoniae, Lactobacillus gasseri, Gardnerella vaginalis, Lactobacillus inners, Proteus mirabilis, and Enterococcus faecalis. This simply means that microbial communities of the 6 symptotypes are distinct based on the presence and abundance of non-E. coli microbiota. In conclusion, there were 6 distinct symptotypes in this population of 225 women with UTI symptoms, characterized by severity and degree of bother of certain UTISA components. The structure of the overall microbial community appears to differ across symptotypes. However, host genetic differences may also account for this difference. These data suggest that the current definition of UTI may encompass multiple distinct disease states, and that E. coli alone may not be the underlying cause of those states. Future research should be undertaken to investigate the role of urinary microbiota structure in UTI development and symptomology. Thank you for your time.